Sloan, and this is AP Biology. Hi, say hi. Hi. All right. So we are discussing, I'm going to get this going here. We are discussing Chapter 14, Biotechnology. And basically, there's just several different processes you need to be able to understand and apply. So let's look through the list right here. Okay. So if each of these you want to be able to like describe the steps of it or the importance of it or the role it plays. So you have recombinant DNA techniques with restriction enzymes and ligase, PCR, gel electrophoresis, biotechnology products, things that are transgenic, bacteria, plants, and animals, gene therapy in and ex vivo, CRISPR technology, genomics and proteomics, and did you do your highly suggested reading and thinking? And then also I just grabbed it right directly from the College Board, the expectations that they listed right here. So maybe you take a quick minute. Are there some there that you need to focus a little bit more on? Do you have a familiarity with it, or is it your friend? You need to know well enough that you can apply it, not an acquaintance, but that you feel really comfortable with it. Okay? Tell your bio buddy you're sitting next to, what do you think you need to spend a little bit more time on, possibly, or maybe you've got it all nailed. I don't know. Tell them. All right. Come back to me. This is biology. Did you know that too? It's biology. Okay. All right. So first on our list is we're talking about recombinant DNA technology, and for that you need to have scissors to cut the DNA. What are scissors? Restriction enzymes. Where do they cut at? A recognition sequence, which is a specific series of bases, right? A specific series of bases. You could have a blunt cut where they just come in and cut across, but better still is when you create overhangs, right? Because then if you cut two different segments of DNA with the same restriction enzyme, you can mix and match, right? You can mix and match in there. Yes?
So the same restriction enzyme cuts both foreign DNA down instead of the bacteria.
I'm not quite there, but yes, if you want to combine DNA, if you want to make a transgenic organism, if you want recombinant DNA from two different sources, you have to cut them with the same restriction enzymes, right? Because then the overhangs will be complementary to each other, right? And once you cut the DNA and you try to combine it, you know what bonds will easily form that we use SSB to keep apart when we're doing DNA replication. Hydrogen bonds will easily form, but what do you need to use in order to seal the phosphate sugar backbone? Ligase for those phosphodiester bonds. So you need to have chemical scissors and chemical tape. The tape is not specific. If there's a broken bond, that'll fix it. The restriction enzymes, specific. Yes? Maybe if you're fragmenting DNA and you're going to do gel electrophoresis, it could. Yeah. All right. And when we talk about the origins, where did restriction enzymes come from? Bacteria. Why did they have genes that would code for restriction enzymes? Why would they do that? Self-defense, part of their immune system, right? To protect themselves from invading viruses, just cut up the viral genome. All right. And where did we get our ligase from that we use in genetic engineering? Viruses. Yeah, they call it T4 ligase, and so they got it from a genome that is located in a virus. Now, there are other ones too, but that's just the common one is the T4 ligase. All right, perfect. Oh, it says right here, ligase from a bacteriophage. Remember, bacteriophages are viruses that infect bacteria. All right, any questions on that? Okay, and I can't remember if I did an overhang page on here. I didn't, so I wish I had. But remember when we look at those, the bases, right, that are complementary, if we cut, then the same bases are exposed, right, and so that you can fuse them together. All right, next, if you want to take a eukaryotic gene and express it in a prokaryotic cell, you have to understand how that prokaryotic cell works, right? And one of the things is they don't have the ability to clean up that gene and remove those intragenic sequences. They don't have the ability to cut out the what? Introns, right? So if you're going to give it to them, you have to give them a clean copy. So what would be a good thing to start with? What kind of RNA? mRNA, and not the primary transcript, right? The one that's already left the nucleus that's good to go. Does that make sense? It's already been cleaned up. And use what? Reverse transcriptase to get a DNA copy, a clean copy with no introns in it. Another enzyme replicase or PCR. You could double strand it. Okay, so now that's what you want to use. Also, when you are wanting to have a bacterium, let's say, express that trait, you have to put it next to what? What do you have to put it next to? A promoter, right? You need to have some regulators. So you need to have a way to turn it on, right? So the best thing to do is just take advantage of a gene it already uses. And remember how prokaryotes are. They have all of their metabolic sequences controlled by an operon. They're all controlled together. So take advantage of that and just sneak your gene right in next to it. So when it turns that gene on, let's say to digest lactose or arabinose, it will additionally turn on your gene right there within that sequence. Okay? So needs to be in a regulatory region and no introns. And then you have to have a way to have it in. And so usually that vector that you're using, right, is a plasmid, okay, that you have cut the same way as you have cut the host genome. Yes? Remember, we're not going to turn anything into anything, right? It is what it is. But it can read the mRNA, and it can bring in a nucleotide that's a DNA nucleotide that's complementary. So if on your mRNA you have a G, then you would, so it would have guanine, it would have ribose and a phosphate right there, yeah? So it would bring what's complementary to G? C. But this time the cytosine would be connected to deoxyribose and a phosphate. It would be a DNA nucleotide. It does the absolute reverse of RNA polymerase. Does that make sense? Okay, what else? Yes.
And then, once it becomes a DNA strand, would it have, something, make another complement?
Yeah, there are other enzymes like replicase and things like that that you could make it double-stranded and with DNA, right? Yeah. What else? Any hands I'm missing? Yes.
So after the genes are put next to, the promoter,
or some other gene that's controlled by a promoter.
And if it's activated, it'll activate the other genes that are related to as well, right?
Yeah. Yeah, you're just sneaking it into a gene that you already have a way to turn on. Just like the lac operon, how you can turn that on with the presence of lactose. But you've just added another gene into that sequence. Remember how we talked about when we looked at TRIP, right? There were 5 different enzymes, 5 different genes you were coding there. So you're just adding another gene in there. So when you turn on that gene to make the enzymes to digest lactose, it just also turns on the one we snuck in there. Okay, what else? Question? You look like you had a question. Do you have a question? You good? All right. So next, this kind of makes a little list of the steps that you need to do. And here you can see those overhangs. And if you cut your eukaryotic gene with the same restriction enzyme as you cut your plasmids, then the bases will be complementary to each other. So they'll form the hydrogen bonds, and then you use ligase to seal it. So then you have your genetically engineered plasmid. You force transformation where it would take up that plasmid, and then it'll express that plasmid just like it would express any other DNA within that host cell. Now, we talked about selecting for those bacteria that were actually successful because you're not going to get 100% transformation. Some of the bacteria you're going to transform and some not. So one thing you can use is you could use a selector as having another gene in here. Do you see where it says AMP kind of right there in green? That stands for ampicillin. So what you would do is on this plate, it would not only have nutrients, but it would have the antibiotic ampicillin. And the only bacteria that could survive are the ones that uptook the plasmid, because the plasmid contains the ampicillin resistance. Absorbing that. Remember we talked about markers so you could know if you were successful. So if it can survive on this plate with ampicillin, then it got your plasmid. If it didn't get your plasmid, it would what? It would die. It would die. Okay, more questions on that? So that's what I put right here. Then plate the bacteria onto medium that control bacteria that would be unable to grow without the plasmid. You have to have the gene for ampicillin resistance. All right, next. Why was archaea really important? Archaea can withstand high temperatures. So therefore, when we do PCR, that's a good source of what? DNA polymerase. Because you're having this elevation and then cooling again and again and again. And normally enzymes would get denatured by that continual elevation of that temperature. The process, don't do anything weird. The process, okay, of PCR is you take your DNA and you're going to heat it up. And the purpose of heating it up would do what? Separate the DNA strands, which normally what would we use to separate the DNA strands? Helicase. Good. And we don't want to replicate all the DNA, just some of it. So what do we use? I'm sorry, I'm not hearing it. Primers. Somebody saying primers out there? Did you say that, Samir? Yeah. Okay. So we're going to use these primers. I don't think you said it. You're going to use these primers to mark because they're going to be complementary to the sequence we're trying to copy, right? So those primers are put down. We need nucleotides. What kind of nucleotides? DNA nucleotides or RNA nucleotides? DNA nucleotides, because this is like a DNA photocopy machine, right? So we have a bunch of DNA nucleotides. What is a DNA nucleotide? It has a what, a what, a what? Base, sugar. What's the name of the sugar? And a phosphate group, right? And we use TAC. TAC is going to then help us line up the correct nucleotide to the original strand. So now we have a complementary strand, right, to each of the original DNA strands. And we talked about that sequence. Denaturing is where you elevate that temperature. Then you cool it down a little bit to allow the primers to anneal on. Okay, and then you can increase the temperature a little bit to facilitate that transcription. You don't have to memorize any of those temperatures. You just need to know that a thermocycler is doing that for you. Okay, questions about that? Okay. All right, gel electrophoresis. What is the purpose of gel electrophoresis? To do what? Sort DNA according to size. Good, according to size. What was the purpose of PCR? Yeah, it's like a DNA photocopy machine. You want to make multiple copies of that DNA. What was the purpose of restriction enzymes and ligase? To make recombinant DNA, to mix DNA from different sources. Okay, so you want to make sure you keep like, what's the purpose of each of these steps? So gel electrophoresis, you're sorting according to size. You are using like expensive gelatin and it has little divots in it. You're loading your DNA in it. It's submerged underneath the fluid and you're turning on an electric current. Whether you cut up the DNA or you don't cut up the DNA, what's the charge on DNA? Negative. So it will always be attracted to positive. So if your well holes are right here and you load your DNA and you turn a current on, it's going to move towards the positive side. Okay? And the assumption is what? DNA that is the same size will move the same distance, right? And so in that way, you can make an RFLP. Do you remember what an RFLP is? Tell your bio buddy what an RFLP is. Restriction fragment length polymorphisms. Do you remember that? So in each class, I'm sure I had a long lost child that I had in each of the classes, right? And we're assuming that that child's DNA would be similar to my DNA, yes? Okay, so they would have the same recognition sequences as I have. So their DNA would be cut up into the same size lengths as me, yes? So if you put our DNA in there, then the DNA would move the same distance and you should see the same banding patterns. And then additionally, right, so that's a who's your daddy or who's your mommy thing. But what else could we do with this? Pardon? Yeah, a crime scene for, forensics. You can use it to look at evolutionary relationships. You can use it for ID of a species, right? So all of those things, that's what you're using, because you're assuming I cut you up in the same size if you're similar, right? If it's the same DNA. Okay, next. This just kind of shows you the process of that, of loading the DNA in, turning on an electric current. You need to stain it with something or so you can view it, and then you can compare it. And for these as well, you always have a control, which is a basis of comparison of known sizes. So you can say, oh, it must be a fragment this big because it lined up with this control fragment. You okay so far? Yes. Contains DNA from two different sources. So I just think of it like recombinant. I hear the word recombine in there. So I recombine DNA from two different sources. Okay. All right. Moving on. So on biotechnology, so you're trying to create some sort of product. The three categories of products we looked at was genetically engineering bacteria for multiple purposes, to produce pharmaceuticals for us, to clean up spills for us, whatever it is that we're mining. Maybe they can help protect plants, make them frost resistant. So that's genetically engineering bacteria. If they contain DNA from a different organism than themselves, they're referred to as being what? Starts with a T. Transgenic. Okay, if you have DNA from a different organism and we've inserted that in. Okay, and then we talked about genetically engineering plants, but plants are not unicellular, they are multicellular. It takes a little bit more work on that because you have to get it into the zygote without destroying the zygote, right? And so that will take a little bit more work, but again, you could genetically engineer plants so that they produce their own pesticides, right? Maybe protect them. And then there's a whole bioethical issue surrounding that as well. Okay, and then we talked about transgenic organisms. And remember when we talked about DNA and we talked about epigenetic factors of control, right? And we talked to things about methyl groups. Do you remember what methyl groups did? They shut it down, right? Made it like heterochromatin, right? And do you remember what helped express traits? Exactly. So when you go in and you're trying to genetically engineer something, you have to be aware that it might already be specialized to that tissue, right? And so if you mess with it there, you might already have methyl groups that'll shut it down. So that's why you might start with an egg, right? Some diploid cell, genetically engineer that. Once you have that fixed, then what you're going to do once you've altered that DNA is find an egg that's, a fresh egg, take out the nucleus, that's called enucleating the egg, put your diploid cell in there, find a host uterus to put it into, and then out comes this transgenic goat that you can harvest your product from. But this transgenic goat, you're not going to have it have sex with another transgenic goat to get more transgenic goats. You're going to have to probably, if it's very specific, you're going to have to do it by hand every single time. Take a cell from this goat, put it into an enucleated egg, put it into a host uterus again and again. Does that make sense? Okay. All right. And then ex vivo, in vivo. When you think X and in, I just think exit and inside, right? That just kind of makes sense to me. So ex vivo, when you're doing that, you're removing those cells. They're exiting your body. You're going to fix them, and then you're going to return them to that body. In vivo, you're not removing anything from them. You're giving them a spray or something, and it's going right in. That would be in vivo. Are you okay with that? I don't think this chapter is as hard as your other chapters, but you're looking at me like it's hard. Are you having a hard time? Just for the day? It's a lot. Okay. All right. Next. Okay. We looked at some of the applications that we have seen. Genetically modified food. If you could genetically modify plants to grow with salt water, which we have more of on this planet than fresh water, that would be really good, right? If you could get a mullet plant where you got, tomatoes on the top and potatoes on the bottom, if we could genetically engineer that, then you could get like multiple items harvested from one plant, right? All of those things would be good. Larger product, larger fruit, whatever it is. The Siri gene, that's just an example of manipulation where you're able to change what would have been a female mouse into a male mouse by giving it that sex determinant gene, which is ultimately what would make it male. The knockout mouse was just a way, like if you wanted to be able to treat something or see if you could fix it, you knock out the good genes and then you have this knockout mouse that's broken, and then you see if you can fix it. Okay, questions about that? All right, next. Okay, we talked about CRISPR. Now, originally, this is also the same kind of thing. When you see these restriction enzymes, it comes out of out of an immune response, right? Of bacteria to kind of protect themselves. But now this has been used. That's what biotechnology does. But what's great about CRISPR is you have the scissors, these Cas9 scissors, but these scissors don't have to be specific. Like when we were talking about recognition sequences, they had to cut at a specific what? Specific series of bases called the recognition sequence, which is unique for each restriction enzyme. This is just a straight up scissor. It gets near DNA. What is it going to do? Cut it. Great. And what you can alter is where those scissors go. What will help you alter where those scissors go? Yeah, the guide. So you can remember we talked about personalized medicine, right? So you can get the guide that is needed to go right to the target in your cells, in your DNA, about what needs to get fixed. And then how they have modified it from its original intent is they can get this Cas9 to stop being scissors. Instead, it will do something else. So bring in some other enzyme in order to facilitate transcription or stop transcription or knock out an entire gene. It is literally a genome editor, right? And the guide tells it where to do that editing. That's the powerful part of it. And it can happen, like when we think of in vivo, it can happen within you. Okay? Right? And then this is just a list of several. It's so rare that this works, but we'll see. Keep going, go over here. This is just a list. Oh, caca. This is just a list of several applications. Is it going to seize now? Several applications that you could review. Sorry, here it is eventually. There it is. Several different practical applications for each one of these that you could review and take a look at. I'm trying to move myself unsuccessfully. Any questions about that list? Are you okay with that? Today is just not a good day. Okay, then when we talk about genomics is talking about DNA, right? And studying and comparing DNA. And so the sequencing that has already taken place, there's literal databases that you can use to compare genomes for like evolutionary relationships, perhaps, or for medicine, like in the HapMap projects, to see what's unique about different people. And then when we look at the structure of DNA, when we look at the repetitiveness of some of them, or these transposomes, and we look to see what we actually express our exons, and there's so few of them, but do you remember when we revisited the central dogma that the intent may not always be a protein, where we talk about the DNA codes for RNA and codes for protein? Maybe the intent of part of that genome is just to make RNA, right? We know we make things like rRNAs and tRNAs, but maybe some of these other RNAs are regulatory in function, and we can't just always think of as protein as our final product. We talked about single nucleotide polymorphisms. what you could remember from that is what kind of mutation? Point mutation. And sometimes it can be horrible and sometimes not so bad, but that is unique between individuals. And then when we look at our genes, that we have the genes are in purple here, and then you have intergenic sequences outside of the gene. Within the gene itself, within the purple, you have your introns and your exons. Okay, and the introns are intragenic within that gene. Yes.
Polymorphism, is that just the different recognition sequences between type neurons?
A recognition sequence, by definition, is a series of bases where a restriction enzyme would cut. Okay, what this is just saying, when you look at DNA, we have variations amongst us. And so if it's one nucleotide difference, then it's just a single nucleotide polymorphism. Yes.
So is the purple intergenic and then the red intragenic?
The purple is the gene itself. Okay, and outside of the gene, that's your intergenic because it's between the genes. Once you grab that gene, within that gene, this is typical of eukaryotes, you have introns and exons, right? Okay, so the introns, which is in red right here, those would be intragenic within the gene, within the purple gene. Does that make sense? Okay, and then what you actually express are those exons. But keep in mind, the combination of exons for one protein, you could get a different combination of, you could eliminate these two here off the end and get a different protein. And that's why we said the proteome is so much larger than the genome, because just by manipulating which of these exons you express, you can change the protein that you're going to get. Okay, more questions on that? Okay. So functional genomics is trying to understand how all of this, yes?
Are intergenic reasons between genes and DNA, are they made of nucleotides or just like different?
Yeah, all DNA is made out of nucleotides. Yeah. Okay, so functional genomics is you're trying to understand the role of everything, whether it be intergenic, you're trying to understand it. And so they do that when they compare different species, closely related species, and then just us. And they make comparisons between it. You can get genetic profiles. It can help you with your personalized medicine of what works specifically for you based on your genome. Okay? Proteomics, it's all about the proteins. And you understand proteins because we've studied its structure. And the DNA for a gene, for an exon, is only spelling out what of that protein? The primary sequence, right? Yeah, that's all you get out of it, right? That particular exon is going to code for a specific series of amino acids to build your protein. But there's more to that. You're going to have alpha helixes and beta pleated sheets for your secondary structure. What else are you going to have? Tertiary structure. And so obviously there are other genes coding for other proteins to make sure that happens, right? So that's what you're looking at when you look at proteomics. Okay, and you can read that if you want to, and then we revisit the central dogma. The way that is expressed all the time is DNA codes for RNA and codes for proteins. Now, it could be DNA is coding for that RNA, and that could be the end all. That could be the purpose of that gene. It's just the RNA that you get from that. And then the last thing I think I have on here, I think I already gave you this, the practical applications. So that's good. And then that brings us to the end. Okay, any other questions? Can you pause just for a minute and listen to me? Some of you came into this review today having already reviewed. And that was really clear. And I think that's the way you want to approach it because you were able to ask those clarifying questions. Some of you showed up today and you were hoping I would reteach you everything that you hadn't taken the time to learn yet. Make these reviews effective by spending a little time reviewing before you come in here. Okay? Yes?